All right, welcome to Introduction to Ecology, lesson number 3. And like the last lesson, today's lesson will be focused on evolution. And again, we're going to focus on only those aspects of evolution that are really pertinent to ecology because evolution is a whole course in and of itself. So in lesson number 1, we learned the definition adaptation. And adaptation is a trait or characteristic of an organism that improves its ability to survive and reproduce. And so the example we learned about was coloration in moths. And so color was an adaptation that allowed gray moths to be more camouflaged and survive better against the sooty trees that appeared after the Industrial Revolution. Now, lesson number 2, we learned about adaptive evolution. And this is a process. So adaptive evolution is the process by which changes in traits that confer survival or reproductive advantage increase in frequency over time. And so again, adaptive evolution occurs because natural selection acts on these adaptations, acts on these good traits, and causes those traits to increase in frequency in a population. And so the gray coloration, which was an adaptation, increased in time in the population until that moth population was almost entirely gray, and that occurred through the process of adaptive evolution. So typically we see variation in adaptations across space. And one good example is monkey flowers in western North Carolina. So monkey flowers grow in 2 different populations. There's a coastal population and there's an inland population. And if any of you have ever been out to California, you know that the coast is very different from inland. So on the coast, we tend to have cool, foggy conditions with high year-round soil moisture. It's just very damp, right? Think about coastal California up in the Big Sur area. If you go even 100 miles inland, you get a very, very different environment. It's very hot, it's sunny, huge components are actually desert, and even in the rest, you get a strong summer drought. And so you have very, very different environments, even though they're fairly spatially close, even though it's all in one state, it's all in the state of California. And so what we find is that the monkey flowers in these 2 different regions are actually very different. They have different behaviors, they have different lifestyles. And so the monkey flowers on the coast bloom continuously from June through November, and then they actually individual plants live for multiple years. They're what we call perennials. And so they sort of have this extended lifespan where they're living many years, they're blooming for long chunks of time. Then we look at the inland population and we find that almost all the flowers bloom before July. And so most of the blooming occurs in a single month in June. And in fact, the flowers, the whole plants, not just the flowers, sorry, the whole plants are dead by mid-July. So they're annuals, they only live 1 year. And so the only thing that's left in July in the inland populations are seeds. There's no adult plants that are still around. And interestingly enough, these 2 different populations, even though they're both monkey flowers, they don't do very well when you do transplant experiments. So if I take a coastal population, if I take a flower from the coastal population and I move it to an inland region, I find that it dies. And it dies because the coastal transplants just bloom too late to be able to reproduce inland. By the time they're blooming and sort of getting started on reproduction, it's already too hot and too dry for a plant to live. If I do the opposite thing, and I take an inland plant and I move it to the coast, I find that it really struggles as well on the coast. And that's because the inland plants waste a lot of valuable growing time because they bloom too fast and die. And on the coast, there isn't sort of this intense sunlight that they can use to gather energy really quickly. It's cold, it's damp, it's not really great growing conditions. So if you try and get your life in too fast, you don't end up doing very well. You don't have enough energy to grow. You don't have enough energy to survive. You need that extended lifespan on the coast in order to make it. And so this is an example where different adaptations are beneficial in different places. And so if we look sort of at the West Coast in general, what we find is that we have these coastal perennial populations all the way up the coast for monkey flowers. And we move just a little bit inland and we have these early blooming annual populations along the inland. Those pink dots are actually inland perennials. And you might say, well, that doesn't make sense. You told me that inland it was too hot and dry too much of the summer and too early in the summer for perennials to make it. They have to be seeds by the time we get to the drought. And the trick about that is that those inland perennial populations are actually near lakes where it's still quite damp all through the year. And so you actually see, even more fine-scale differences in adaptation when you look inland. There are the inland annuals, which is most of the plants, and then around these lakes you actually have inland perennials that are more similar to the coast. So that's adaptation, and what you see is we have sort of different populations in one place versus another. But sometimes you actually get a gradual change in frequency of an allele or in the sort of average characteristic of a trait over a population range. And when this happens, we call it a cline. So one really good example of a cline is moose size in Sweden. And so if I took a look at moose in the south of Sweden, they tend to be very, very small. When I go to the middle part of Sweden, they tend to be a little bit bigger. And when I go to the far north of Sweden, where it's very, very much colder, I get very, very big moose. This is actually a broad phenomenon we see across a lot of animals in a lot of different places. Animals tend to be smaller in the south and bigger in the north, even within the same species. And so this is a cline. And this is an example of a cline where it's a characteristic. It's the size of the animal. So that's sort of a physical trait. We can also have clines in the frequency of an allele, and a good example is in the human population. So again, if we take a look at warmer areas versus colder areas, people exist across all of those, right? We have people all the way from the equator in Africa all the way up to sort of not quite the North Pole, but pretty close up in Finland. And what we find is that there is a TRPM8 gene that allows people to sense cold. And it's actually at this particular locus called the RS1016694 SNP, or single nucleotide polymorphism. And so people who have a C at that allele can sense cold better. And what we find is that the frequency, people with a T can sense cold better than people with a C. And what we find is that the frequency of that T allele, the frequency of that sort of trait that confers an ability to sense cold, increases as you go from Africa to Finland. So maybe it's not too important to sense cold in Africa because it's never cold, but it's very important to sense cold up in Finland, at least historically, so you know, hey, I got to get out of the cold. I got to start preparing for winter. I got to start going inside so I don't get frostbite. And so what we actually look at, if we take a look at the frequency of that T as a function of latitude, you can see it kind of just goes straight up all the way up. And I have largely northern heritage, so you know, my grandmother came from Norway. I have a lot of family from Northern Europe. And interestingly enough, if I look at that particular SNP, or that single nucleotide polymorphism in my own DNA, I have 2 Ts, which shouldn't be surprising. I'm from a more northern location than a lot of people in the world. So again, that's a cline, but instead of sort of a gradual change in a trait, instead of sort of a gradual change in size or something like that, gradual change in color, it's a gradual change in the frequency of a particular allele or a particular gene form. Now, we all know, or hopefully we all know, that climate change is happening. And so what's interesting is that some of these clines that are associated with temperature, whether it's size in moose, and the bigger you are, the better you survive in cold weather, or whether it's actually frequencies of alleles that allow you to sense cold or allow you to deal with cold, what we find is that those seem to be changing and have been changing over the last 50, 100 years as climate change has been ongoing. And so if I take a look at a gene that confers cold tolerance, the ability to withstand cold temperatures and live through cold temperatures, you might have a cline where, you know, down in Mexico, only 10% of the population has that cold tolerance allele. By the time you get to, I don't know, Maryland, in that area, Iowa, about 50% of the population might have that cold tolerance allele. And when you get up to Hudson Bay or further north, up, you know, the northern parts of Canada, 100% of a population would have that cold tolerance allele. Well, now what we're finding is that with climate change, you know, we're getting the population shifting. So even in Virginia, maybe it's only 10% of a population that'll have cold tolerance. And to get 100% cold tolerance, you got to go all the way up to Greenland. So we find that the climes are actually shifting in response to warming conditions across the globe. So that's spatial variation in adaptations and how different environments, different locations can actually select for different traits in different places. We can actually have temporal variation in adaptation as well. And a good example is in brassica, which is sort of related to broccoli. It's a type of plant. And there were some brassica rapa that were growing on out in California again. And in the 90s, there was a lot of rain that happened in this particular area where they're growing out in California. And, all the way through until about 1997, 1998, up until the early 2000s, it was a pretty wet environment. But then between 2000 and 2004, there was this multi-year drought. And so that put a very different pressure on those brassica plants than sort of the environment they've been living in the 90s. And so scientists actually had fortunately gone in and done some studies on these plants in the 90s, and they went back after the drought to see if anything had changed. And what they found is that, before the drought conditions, the flowers were blooming in June through July. And again, because they didn't really have to worry about getting that blooming done before the drought hit. They didn't have to worry about that so much because there was no drought. It was pretty wet most of the time. But then when they went back in after the drought, they found that the flowers were blooming 2 to 9 days earlier. And the reason they were doing that, similar to the monkey flowers, they wanted to get, the seeds set. They wanted to get sort of their whole life cycle done before it got too hot and too dry in the summer. And so again, what had happened was the plants that just naturally bloomed earlier, they had an advantage. They had an adaptation that allowed them to survive and reproduce better in that drought period. And so you actually had the population shift so that you had more plants that were blooming earlier and less of the plants that were blooming later. So again, that's temporal variation in adaptations. So you may not have noticed it, but we've talked a lot about evolution, and evolution can occur in response to both abiotic and biotic conditions. If I take a look at my monkey flowers and when they flower, or I take a look at my moose and the size of my moose, those are determined by abiotic conditions. For the monkey flowers, it's water availability and to some extent heat. For the moose, it's temperature. Those aren't living things that are sort of governing it. Those are abiotic conditions, water and temperature. On the other hand, if I think about the crickets on the Hawaiian Islands, and you remember they stopped singing in response to the parasitic flies, or if I think about the human population and selection for or evolution of the ability to drink milk, again, that's because of a species interaction. That's because of our tight coupling with cows. And so those are basically evolution that's occurred in response to biotic conditions, to predators, to parasites, to food source, to anything like that. And so from both of those examples, we actually see how ecology influences evolution. If you remember back to the first lesson, ecology is the scientific study of interactions between organisms and their environment. And so interactions between organisms basically affect evolution through things like parasitic flies selecting for very quiet crickets on Hawaii. And there's many other examples of that where it's sort of that tight coupling between two different species that drives the evolution of one or both of the species. However, ecology is also the study of interactions between organisms and their environment. And so again, it's an ecological interaction that drives evolution of monkey flowers in the two different locations in California. It's that interaction of the monkey flowers with the water availability and moisture and precipitation that causes evolution of an annual versus perennial flowering time, and that different sort of early season versus throughout the whole season flowering and seed setting. So ecology influences evolution, but the same in reverse is also true. Evolution also influences ecology. So if you remember, one definition of evolution was change in allele frequency, and the other was descent with modification. And so you can imagine that, if I have plants and they're getting eaten by herbivores, those plants may be selected to change the population. So if I introduce a herbivore that's eating that plant, then that might select for a change in allele frequency in the amount of an allele that produces a toxin. Alternately, I might have the plant evolve a new toxin or multiple new toxins, and then I might get sort of plants that are much more toxic, and that's sort of descent with modification. But either way, when the plants evolve a toxin to prevent herbivory, they're basically influencing an interaction between themselves and the things that eat them. So they're influencing an ecological interaction. They're trying to prevent insects from eating the plants, and that's an interaction between two different organisms. So that's an ecological interaction. So evolution also influences ecology. And that brings up a really interesting idea, this idea of coevolution. But whenever evolution is driven by two interacting species, neither is static. Both populations, both species, are undergoing changes in response to natural selection, right? And so what you end up with is essentially an arms race where you have coevolution, where you have reciprocal changes in the two species that are interacting. And a classic example is, again, with plants and toxins. So the plants might evolve a toxin, and that then produces the herbivore, in this case a grasshopper, from eating it. The grasshopper might then evolve some metabolic pathway to break down the toxin that the plant made. And that's coevolution. They're evolving in response to each other round and around. And so what does the plant do? The plant evolves a new toxin that the herbivore does not have a metabolic pathway to break down. And we go around and around this cycle over and over, and that's called coevolution. So evolution seems like a pretty good system. You know, it makes things kind of match their environment, and it makes things match each other in terms of species interactions. So you might wonder, why is anything ever not perfect in nature? And the answer is that there's always limits to perfect adaptation. This is a great picture by Gary Larson, Far Side. This one always cracks me up. But basically, it's an example of imperfect adaptation on the part of the dinosaurs, who are busy making fun of the mammal as the snow comes. So, why don't we have perfect adaptation? Well, one reason is gene flow. Another reason is environmental change. That's the case with the dinosaurs. A third reason is lack of genetic variation. Remember, all of your alleles come from mutation. So if mutation doesn't happen to make that perfect allele, you can't sort of cause the frequency of that allele to increase in the population. That's all that natural selection can do, right? Because it doesn't exist at all. We also have evolutionary history, and that puts on constraints. And then we have ecological trade-offs. And again, that's an issue with constraints. So let's take a look at each of these in a little bit more detail. So gene flow, we talked about gene flow in the last class, and we gave this example with the GMO fields and the non-GMO fields. And we said, well, you know, the organic farmer is not really happy when the pollen floats across the fence from the GMO field into the non-GMO field, because now he's got problems selling his crop as a non-GMO crop. But the reverse is also true. It's true that some of this non-GMO pollen can float into the GMO crop, and it can cause some of those plants to not have those genetically modified alleles. And frequently, what they're doing with genetically modified organisms is they're making them resistant to pesticides. So when Mr. Organic sends his pollen over and makes these non-resistant plants in the GMO field, and Mr. Non-Organic waters his field with pesticides, it's a problem because some of those plants are not resistant to pesticide. It kills them at the same time as killing the weeds. And so, you know, that's a problem. It's a problem for both farmers. It's a problem for the farmer who wants to be non-GMO because he can't keep the GMO stuff out, and that makes a problem selling his crops. And it's a problem for the GMO farmer because he can't keep his GMO fields purely GMO, and so he loses some plants when he sprays them with pesticides. And so it's because of this gene flow that the GMO and non-GMO corn isn't perfectly matched to the GMO plot and the non-GMO plot. We've got some non-resistant corn in the GMO field that we're spraying, and we've got some resistant corn in the non-GMO field that we want to sell as organic produce. Same kind of thing with the natural world. So just like farmers, natural selection has trouble perfectly matching one trait to a particular location when there's gene flow from another location where conditions are different and things that are adaptive, those good adaptations are different. So again, another plant example, this time in a natural system, there are sunflowers. And sunflowers can grow on serpentine, which is sort of a very unique chemistry rock. And they can also grow on non-serpentine soils, right? And so you kind of need special traits to grow on these serpentine soils because they're very nutrient poor. But we find that populations of serpentine sunflowers actually grow on both areas. Now, if I take a non-serpentine plant and I try and move it to the serpentine field, most of the time I find it doesn't do very well. And that's because it doesn't have the ability to extract nutrients from that very nutrient-poor soil. I do the same kind of thing. I do another transplant experiment, and I take a serpentine sunflower from a serpentine field and I try and plant it in a non-serpentine soil. And I find that, it survives, but it just does not produce as much seeds. So again, survival may not be compromised, but reproduction definitely is. So it's not particularly adapted to the non-serpentine soils. All right. But the interesting thing is that when I take a look at serpentine and non-serpentine sunflowers, I do see this local adaptation. I see that I take plants from one field and they don't do well in the other. I take plants from the other field and they don't do well in the first. But they both share genes from each other. So I see some non-serpentine genes popping up in my serpentine field, and I see some serpentine genes popping up in my non-serpentine field. And the reason I see that is because of gene flow. So the adaptation persists. The plants that grow in the serpentine fields are predominantly serpentine, and they mostly have serpentine genes that allow them to do well, and they don't do well when you move them. And the plants that grow in the non-serpentine fields have predominantly non-serpentine genes, and they do best in the serpentine fields, and they don't do very well when you move them. But I do see this crisscrossing of a few different genes, a few of the alleles from the serpentine field end up over in the non-serpentine field, and vice versa. So adaptation persists because there's really strong selection that causes the plants to die or just not produce seeds when they're moved, but we still have a single species there because they continue to mate with each other. We continue to get genes swapped between the two. And as a result, probably those non-serpentine flowers could do even better if they could just get rid of those serpentine genes that keep flowing in. And the serpentine flowers could do even better than they're doing if they could just stop getting that garbage from the non-serpentine flowers. But they can't because the pollen just keeps blowing between the fields. They're frequently serpentine and non-serpentine areas are close to each other, and so the wind just blows the pollen every which way. So another reason why you don't have perfect adaptation is environmental change. And again, that's that example with the dinosaurs laughing at the mammal. So sometimes you have historic conditions that are different from current conditions. And because it takes a while for natural selection to occur, you don't necessarily have perfect adaptation because some organisms are still trying to get caught up to the change that happened. They were good, and then something happened and they have to move and find a new moving target. So one good example, again, with climate change is the great tits in Britain. So historically, the cues that the great tits used to decide to start laying eggs aligned with the same cues that the caterpillars used to decide to emerge from their eggs. So everything was great. When the caterpillars appeared, the great tits were, laying their eggs. They had a lot of energy needs because they had to feed all those babies, and it was all nice and perfect. Now, unfortunately, even though those two cues that the two different species used aligned, they were not the same cue. And so, you had a match just sort of by chance because the two cues matched up. With climate change, however, the cues that the great tits use have changed, the timing of those cues have changed relative to the timing that the caterpillars use, the timing of the cues that the caterpillars use to decide to emerge from eggs. And so what happens is that now when the chicks appear, when the chicks hatch, they actually have no caterpillars because the caterpillars are using cues that occur later. And so caterpillars emerge later. Great for the caterpillars because there's less eating them. Not so good for the birds because they have no food. And so this is an example where we've had a change over time in the environment. And as a result, we have this mismatch between the birds and the caterpillars. And the birds basically need to evolve to lay their eggs later in order to catch up to the caterpillars. They haven't done that yet. Whether that happens before they go extinct, sort of that's one of the big questions in climate change. Another reason why we don't have perfect adaptation is because of a lack of genetic variation. So again, remember I said all alleles emerge from mutation. And mutation isn't a smart process. It's actually a mistake, right? Mistakes happen, new things occur as a result of those mistakes, and sometimes they're happy accidents, just like Bob Ross. And so sometimes biology can use those to its advantage. But if those particular mutations aren't around, then you can't select for them. And so a good example is antibiotic resistance. And this is a nice picture where on the left hand it shows when the antibiotic was introduced, and on the right hand it shows sort of a timeline for when resistance was first identified. And what you'll notice is that sometimes it takes quite a while between introduction of the antibiotic and the emergence of resistance. And that's because the bacteria sort of need to invent by chance, invent through mutation, some resistance pathway that helps them deal with that antibiotic. And that takes time. You just gotta wait until an accident happens to be, the right thing for the right place. And so what you'll notice is that, we're waiting years and years sometimes for bacteria to invent the antibiotic resistance. And bacteria have really quick lives. E. coli lives for about 20 minutes and then splits. So you got a lot of opportunities for mutation to occur. They also have some of them have quite high mutation rates just at baseline. And so, the fact it takes them years to find a new solution shouldn't surprise us that some other organisms take, hundreds, thousands, millions of years. So again, it can take many years for antibiotic resistance genes to appear because they have to be invented by mutation, or else they might have to be introduced from elsewhere. So, if I've been using a particular antibiotic in Europe for years and years and years, and then I start using it in America, it's not necessarily true that the bacteria in America need to invent resistance. All it takes is for one person to bring a bacteria from Europe with them, and now we've introduced that resistance gene, and the North American bacteria don't need to invent it. They just, either borrow it through horizontal gene transfer from the European bacteria that got introduced, or the European bacteria just take over and drive the North American ones extinct. One way or other, we get antibiotic resistance in North America. So another good example where lack of genetic variation was a problem was with wine growers. And so in the 1800s, there was a strain of mildew that was introduced from America to Europe. And unfortunately, this strain of mildew hit the wine grapes. And of course, if you've ever been to Europe, that's very, very important, depending on the part of Europe, but the wine is paramount. And so, what the farmers in Europe could have done, they could have said, well, we're just going to sit back and let our crops die, and we're just going to hope that some plant somewhere ends up with a mutation that protects it from mildew, right? But that could have taken thousands, millions of years, and the mildew could have wiped out most or all of the wine grapes before the wine grapes figured out how to deal with it, before mutation sort of, introduced a beneficial gene. And so instead of waiting, because of course wine is important in Europe, what the Europeans did was they took the European grapes and crossed them with North American grapes that already had resistance to this mildew. The North American grapes had sort of co-evolved with the mildew in sort of this escalating arms race, and every time the mildew found a way to attack the North American grapes, the North American grapes found a way to avoid it, back and forth. And so what they did was they took those resistance genes that the American grapes already had, and they introduced them into the European population. And that's because the European grapes, again, did not have a gene for resistance, and the farmers didn't want to wait and hope that they would invent it through mutation. So another reason why we don't have perfect adaptation is evolutionary history. Natural selection works on traits that are already present. We typically don't a single mutation can't take us from having an arm to having a fin, right? There's a lot of little changes that happen all the way along. And so, same thing, it can't take us from having, one single happy accident doesn't take you from having lungs that breathe oxygen to gills that extract oxygen out of water. And so these small scale changes, you kind of have to rely on a little bit of change at a time. You have to be patient. And so as a result, frequently, we can't suddenly become perfect because we have to work with what we've got. And a good example is with cetaceans, things like whales, dolphins, all of those marine mammals. They still breathe air. You might think, why, if you're a whale, do you want to keep coming up to the surface to get air when, there's oxygen in the water? You just got to invent a way to get it out, just like the fish did. But the fact is, because dolphins and whales and all those marine mammals came from a terrestrial background, it's really hard to suddenly make gills, and so they've evolved in a different way. They've evolved where they have to come up to the surface, and maybe that's not perfectly beneficial, but it works for them, and so that's why they can't breathe underwater. The final limit to perfect adaptation is ecological trade-offs. And we talked a little bit about this when we talked about no free lunch, and we looked at sort of the number of children you have versus their probability of surviving. And we found that, if you have a lot of kids, their probability of surviving may not be so high. You're just like, I just hope one gets through. Whereas if you have only 1 or 2 kids, then you have a much higher probability of surviving because you put all your energy into that, keep making sure that one kid lives. And so we have a lot of different trade-offs. We can see vegetative versus reproductive growth trade-offs. So plants can invest in making leaves and capturing energy and growing to bigger plants, which might be good for competing out neighbors. But if they do that, they don't put their money into, or their energy into reproduction, into making lots of seeds. So instead, they could be little and be like, hey, I'm going to get outcompeted in a year, but I'm going to make bazillions of seeds that spread elsewhere where maybe competition isn't so strong. Another good one in plants is sexual reproduction versus clonal reproduction. So you can put your energy into seeds where you've actually got a mating process with sort of male and female coming together, and that creates more genetic variation, so that can be good in environments where you want to evolve rapidly. But you could also put your money into, instead of making a lot of seeds, you could also put it into clonal reproduction, where you just lop off a piece of the plant and it grows a new baby. It's not as good in terms of sort of genetic variation, but it might be good in a more stable environment where you don't have to worry about sort of being variable to try and find the right place, to try and fit in, to try and evolve to be better. And so, again, you can't do both. You can't have a lot of clonal reproduction and a lot of sexual reproduction because you just don't have the energy to devote to little pieces to lop off for clonal reproduction and making seeds. Another good example that people have shown in red deer is reproduction versus adult survival. Female deer that have more offspring in a particular year are less likely to survive through the winter because they use their fat stores to make babies rather than to store up to sort of prevent starvation when there wasn't as much food. And again, we also have things like fecundity versus juvenile survival, which we see in people, where we have your probability of the child surviving versus the number of kids you have. The fertility, or the number of kids you have, is fecundity. Another good example of an ecological trade-off, here's a little bit more specific. There are southern field crickets, and they can take on 2 different phenotypes. So we can have a winged phenotype, and the winged phenotype, as you imagine, puts all of its energy into wings, so it invests in energy resources to fuel flight. So that means it can go to new food sources, it can fly all around, it can find mates. It's really good in a patchy environment where walking might get you in trouble because you might go from a good place to a bad place and might not be able to get back. And so you see the wings in that particular picture. We also have wingless phenotypes, and they have very, very, very tiny wings, but instead of investing in these big wings with like strong wing muscles, they put all their energy into ovary size and making eggs. So here we have a trade-off between dispersal and the ability to find food and your fecundity, basically, your ability to have more children. And again, you might think, well, why don't they just do both? Why don't they have big wings and big ovaries? And the fact is, it's a trade-off. You can't have both. You can't spend all your money on beer and still have money left over for textbooks, right? You gotta decide what's important to you in your particular situation. And again, that limits how perfectly organisms can be adapted because they just can't be good at everything all the time, all at once. Right. So that's adaptation. And so frequently we think of evolution in terms of a single species adapting to a new environment or adapting to a different environment that occurs at a different point in time, whether it's because of climate change, whether it's because of sort of a short-term drought, whatever. But evolution over longer time periods also leads to what we call speciation. And this is the process by which one species splits into 2 or more species. There's a couple different mechanisms that give rise to speciation. Genetic drift, genetic divergence, and hybridization. So with genetic divergence, what happens is different organisms end up in different places with different environments, and those environments select for different traits. They select for different adaptations in the different places. And as you build up all those different adaptations, you end up with two populations that are so different, they're no longer the same species. They can no longer breed with each other. And so one example is pitcher-wing fruit flies, again in Hawaii. So occasionally initially flies were introduced into Oahu. And they were introduced in two different time points. And then occasionally those flies get blown to another island where conditions are maybe slightly different and they're selected for different features. So we know that the flies have been blown from Oahu to Molokai and then down to Maui. And what we found is that each of these different islands actually has a different species of pitcher-wing fruit fly. But we know that they all came from that same introduction event, or those same two introduction events on Oahu. But what happened is when the flies got blown to Molokai, it had different sort of environmental conditions, it had different biotic conditions, and those selected for slightly different features, particularly in the wing coloration, and that affected their mating because they don't want to mate with things that look different. And so basically what we've got are these different species evolving across the different islands because of genetic divergence. Genetic drift is a similar process, but it's different. So with genetic drift, it's not because the environmental conditions are different and the environmental conditions are driving this divergence between two different populations. What happens is the environmental conditions can be completely the same, but if we have small populations in two different places, then just by chance, those populations end up having different alleles in them, right? And we talked about this again on islands with the Channel Island fox. It's not the Channel Islands, unlike Hawaii, the Channel Islands are all fairly close to each other. They're fairly similar environments. There's no obvious biotic or abiotic differences. And so we assume that a lot of the differences between these foxes is just because, the first couple foxes that colonized one island had a different set of traits than the first couple foxes that colonized another island. And because they were small populations, some of those alleles, even if they came with the same alleles, some of them went extinct on one island and some of them didn't on another, just by chance, remember that process of genetic drift. And so again, the population of foxes on these islands are very small. That allows for random fluctuations in allele frequency, presence, absence, to lead to very different populations on each island. Now the end result with genetic drift is the same as the end result with genetic divergence, whether it's the environment driving the populations apart or whether it's just these random chance happenings driving the populations apart. Eventually the populations end up so different that they don't breed with each other. They're classified as different species, and we say a speciation event has occurred. The final example is kind of rare. It's a weird one. It's not really common. It's not really one of the more important forms, but it's hybridization. And this occurs when we have different species living in different areas, but occasionally they breed in a region of sympatry, and that leads to a third species. So one example is the Lycinid melissa, which lives in the Great Basin desert on the east side of the Sierras. And then we have the Lycinid itis, which lives on wet meadows on the west side of the Sierras. And you can see those in the dark blue and the yellow. And then there's this region where they sometimes meet, and occasionally they interbreed. But when they interbreed, they make a new species, and that new population does not live either in the dry desert or in the wet meadows. It actually lives in an alpine habitat, just the combination that makes it survive only in the alpine habitat. And because it lives only in the alpine habitat, it doesn't run into a lot of Lycinid itis or Lycinid melissas, and so we classify it as a separate species because it's sort of genetically different enough. It's evolved there were a couple of matings of the two parent species in the past, but that third offspring species has evolved and changed enough from the two parents because it's not interacting that it's now a third species. So again, because they don't cohabitate and because the offspring does not mate with either parent species, it becomes a new species that sort of adapts to that alpine habitat different from either of the parents. Again, it's a very rare example. There's not too many cases where that happens. There's a couple. All right, so that's speciation. And you'll notice with speciation, I gave a couple of examples, and one of them was Hawaii, which is islands, and one of them was the Channel Islands out in California, so again islands, and another example involved the Rocky Mountains separating or the Sierras separating an east side and a west side. So you might think, Is that just chance? Does Dr. Bewick just like islands and mountains? No, it's not. And that's because speciation requires separation. So we talked early on in this class about gene flow. We talked last class about gene flow. And gene flow kind of homogenizes populations. It mixes everything up. And so if you have a lot of gene flow, it's really hard to get speciation to occur. Just like it's hard for the GMO farmer to keep his GMO crop purely GMO, and just like it's hard for the serpentine sunflowers to keep their population entirely serpentine genes, speciation is really difficult when you have too much gene flow because you can't get the populations different enough in one place versus another because they keep getting traits from each other. Even though those traits may not be adapted to the locations where they're kind of drifting into, they're still in that population because of gene flow. And so again, that sunflower example is a really good example. These are still the same species because they're still breeding with each other. We're still seeing genes from one population appearing in the other population and vice versa. And so despite their phenotypic differences, but despite the fact that you take plants from one area and they don't do well in the other, these are still a single species because we still have enough interbreeding with the gene flow, even though it results in, some of those sunflowers that end up in the serpentine soil dying because they have too many non-serpentine genes. And even though it means that some of the plants that are in the non-serpentine soils don't breed as well because they have too much serpentine genes, even despite that, we still classify these as a single species because they still continue to interbreed. All right, so that picture wing fruit flies, that was a different example because the islands were separated enough that, the number of fruit flies blowing from Oahu to Molokai or from Molokai to Maui is very, very small. It happens maybe, once every 100 years or something. And so you don't have that heavy gene flow mixing the populations, and that allows speciation. Islands are often cases where you get speciation. And as a result, islands often host a large number of endemic species. What that means is species that only exist on that particular island. And that's because the island is isolated enough from both the mainland and from other islands that speciation occurs readily because we don't have gene flow, we don't have mixing. And so either because of genetic divergence or because of genetic drift, you end up with speciation occurring fairly easily because you're not getting all these genes from other places. Another good separation is anything like a river or a mountain range or a canyon. Again, because the ability of certain organisms to move across that, especially organisms that don't fly or that aren't pollen, is limited. A good example is in the Grand Canyon, where we have two different species of ground squirrels on the north rim versus the south rim. And again, it's because the squirrels don't fly, they don't drift on the breeze, so they don't have a good way of crossing the Grand Canyon. And so because they've been isolated, because we don't have a lot of gene flow between the two populations, we actually have different species. Speciation event has occurred. So frequently we think of speciation occurring because of separation due to physical barriers, whether that's oceans between islands, whether that's canyons or mountain ranges between populations on either side. But we can actually have other forms of separation. And one good example is host plant separation. So if particular organisms, a lot of flies will breed and mate and lay their eggs on host plants. And so if different flies use different host plants and they really prefer one versus the other, you can actually get speciation. That's particularly true if the fruits that they like to use are around at different times in the year. So a good example is in, again, another type of fruit fly, where we have different species on different kinds of fruit. And they're all related. They all probably came from the same ancestral species. But we have one species on apple, another species on hawthorn, another species on snowberry, another species on dogwood. The list goes on. Another species on blueberry. And that's because, if you mate and lay your eggs on a particular plant and you really like that particular plant, then you don't tend to sort of meet and you don't mate with the populations that are on a different plant. So if you're apples and you're like, I go for apples, I only mate on other things when I can't find apples, then you're going to mate with all the apple flies. And if you're only going to mate on hawthorn, unless you just can't find a hawthorn, then you're primarily mating with hawthorn flies. And so that separation, because we don't have gene flow, if we don't have mating, we don't have gene flow, that basically allows for speciation. That's, again, especially true when the timing is different, because then you also have not just the host plant being different, but the timing being different, and your whole life cycle gets sort of split and you allow for speciation. Again, another example where you have sort of this separation is with a hybridization. when you're using different habitats for your breeding. And again, here we had the offspring of the hybridization. It uses alpine habitats, and so it never runs into either parent. If that offspring ran into both parents because it was happy to breed anywhere, it was okay breeding in deserts and in wet meadows, then we wouldn't have had this hybridization resulting in speciation. But again, it's that separation. You need that separation, whether it's due to, again, physical barriers or whether it's just due to using different habitats, like using a desert, using a wetland, or using an alpine meadow. So when we talk about evolutionary trees, evolution, we typically represent it as an evolutionary tree. And this is a branching diagram that represents the evolutionary history of a group of organisms. And so if I look at those picture-wing fruit flies on Hawaii, what you'll notice is that I can draw the speciation events. They all originally came from the same ancestor, and then there was a split. And then, the fruit flies that first colonized Oahu, they split further. And so there was a split when there was a colonization of Maui. There was a split when there was a colonization of the Big Island. And each of those splits is drawn on this evolutionary tree. Same thing, we had a second colonization event of Oahu, and that population split again when we colonized Molokai and the Big Island. All right. So with the picture-wing fruit flies, all of those fruit flies are still around. They all still exist. They're all still hanging out in Hawaii. So it's pretty easy to understand the tree. It becomes a little more difficult when members of the trees go extinct. And so a good example is the primate evolutionary tree. So, chimpanzees are still around, bonobos are still around, and humans are still around. But all those other things in the middle have gone extinct. They're not around anymore. We have fossils, but we don't have them. And so, sometimes you look at chimpanzees and humans and you're like, wow, that's really different. I don't see any evolutionary tree putting those two things together. But the reason it looks so different is because you're missing all those intermediate steps. You're missing all the sort of branches that connected the two leaves. All right. So we talked a little about speciation. We just talked about extinction and how that kind of confuses evolutionary trees. But when you think about it, extinction and speciation are sort of two ends of the spectrum. There's sort of two opposing processes. Extinction removes species, and speciation makes new species. And so when the rate of extinction equals the rate of speciation, it's kind of like the money you make being equal to the money you spend, you're at what we call an equilibrium. We have a stable number of species. Some of them are disappearing, but new ones are replacing those. It's not that we always have the same species, but it's that we have the same number because extinction and speciation balance. When we have less extinction than speciation, then what we're going to have is increasing biodiversity because we're removing less than we're actually adding. So we get more and more added, and we're not taking away that much. The other end of the spectrum, when extinction rates are greater than speciation rates, we have decreasing biodiversity. It's again, you're taking more money out of your bank account than you're putting in. Eventually, you're going to go bankrupt. And that's unfortunately the case we're in now. So that plot on the right-hand side, what you see is the number of different freshwater species, marine species, terrestrial species as a function of time from the 1970s to 2000. And what you'll notice is that all of these different groups of organisms are declining. And so basically what that means is that in our planet, our world today, extinction rates are higher than speciation rates. And unfortunately, that's largely because of us. So mass extinctions have happened in the past, and they haven't been because of us. We had the end of the Ordovician, where 60% of marine invertebrates went extinct. We had the end of the Devonian, where 50% of marine invertebrates again went extinct. We had the end of the Permian, another marine extinction event. 82% of marine invertebrates went extinct. We had the end of the Triassic, 53% of marine invertebrates went extinct. And the end of the Cretaceous, 47% of marine invertebrates went extinct. Unfortunately, we are now at what scientists believe is the 6th mass extinction. So you see there have been 1, 2, 3, 4, 5 in the past, not caused by humans. Now we are probably at the 6th mass extinction. And in this case, as opposed to all those other cases where there are different events causing them, this case, it's us. All right, so mass extinctions are bad. They're particularly bad for the organisms living around now because they're going to go extinct. And that includes humans, right? We're going to have a problem when we lose these species that we depend on. We depend on trees for oxygen, and the Amazon is burning. We depend on crops for food, and with more drought conditions, those are going harder and harder to grow, and we're not going to have them, right? But the good news is that when there are mass extinctions, there's another process that kind of tends to compensate. And so there's this process called adaptive radiation. And this is an event in which a group of organisms gives rise to many new species that expand into new habitats or new ecological roles over a relatively short period. And so what we found is that over a lot of those past mass extinctions, we found an adaptive radiation event that follows. And the reason is that when you wipe out a ton of organisms, there's a whole pile of niches, there's a whole pile of roles for these new organisms, the ones that survive, to fill. And so they radiate into all these different forms that fill these roles that were originally occupied by other organisms that went extinct. So again, we can expect that with this sixth mass extinction, we may have an adaptive radiation event where some organisms, maybe it's cockroaches, I don't know, that live all the way through that extinction period end up having this wide new open space to fill, and they fill it by evolving different forms to fill all the different roles that this new space opened up. Now, adaptive radiation doesn't only occur in response to a mass extinction. It occurs in response to a couple other things as well. So one is a colonization event. When animals were first introduced to the Caribbean islands, what we found was that there was nothing like them in that area. And so there were a lot of different niches, a lot of different roles for them to fill after that first one arrived on the island. And what we found is that there's a whole pile of different forms of them that took place. We had a rapid, sort of a rapid evolution of different types of animals across these islands after the first arrived. And so arriving in a new location can lead to rapid speciation to fill the niches that were never occupied in the first place because that whole entire type of organism didn't exist in that area. We can also have adaptive radiation in response to novel innovations. And so, if you have a fundamentally new behavior that evolves, something like going from land terrestrial dinosaurs to flying birds, then again, there's sort of that one innovation, that one new behavior opens up a whole new range of roles that you can fill. You can be hummingbirds that are tiny and eat nectar. You can be albatrosses that are huge and soar over the ocean. You can be herons and storks that sort of fly to wetlands. You can do a lot of different things once you evolve the ability to fly. And so again, when a novel innovation appears, certain novel innovations, they allow access to a range of new habitats, not just a single new habitat. And as a result, you again get this adaptive radiation where birds kind of exploded once the ability to fly was evolved because there were all these niches suddenly that they could fill. And so different populations filled each of the different niches and we got a ton of speciation events. All right, so that's the second lecture on evolution. And again, as always, if you have questions, email me at sbewick@clemson.edu and I'd be happy to talk to you about evolution or anything else that we've talked about.